his committee's here, so we can go ahead and get going. Connor is in the master's program. He is a graduate of Oregon State University and is now in our program. In addition to his bioinformatics work, he also does some work with Eilish Boudreau and Annette Totten on a project related to telemedicine at the VA. So we'll let him get started.
All right. Well, thank you everyone for coming this afternoon. As Dr. Hirsch said, my name is Connor Smith. I'm a master's student here. So today I'm going to be talking about differential methylation in the context of somatic mutations in AML. So I'm going to start out with some background info real quick on just AML and DNA methylation and everything like that. So acute myeloid leukemia is the most common adult leukemia, acute leukemia. There's in 2018, there's an expected, they expect over 19,000 new cases and about over 10,000 deaths due to AML in 2018. AML is a cancer of the blood and bone marrow, and it's caused by abnormal growth and differentiation of the hematopoietic stem cells, which include the myeloid stem cells and the myeloid blast. These cells are both precursors to red blood cells, platelets, and white blood cells. When immature versions of these cells gain some type of advantage over healthy cells, they start to grow and replicate out of control. On average, a patient undergoing chemo will incur on average over $280,000 in medical bills, and that more than doubles to over $544,000 when patients undergo stem cell transplant. So as far as the genetics go, AML actually has been found to have fewer genomic mutations than most solid tumors. A study of 200 patients found that an average of only 13 mutations were found within genes, and with an average of 5 being in genes recurrently mutated in AML. Of particular interest of these genes, and this is an overall study of mutations that were found in different genes of AML, ranked based on how prevalent they were. Of interest here are the second and third ones, DNA methylation and chromatin modifiers. We're going to start by talking about DNA methylation and move on to chromatin modifiers afterwards. So both DNA methylation and histone modification involved with regulating DNA access and transcription, but in different ways. As I said, we're going to first start with DNA methylation and then move forward to histone modification afterwards. So what is DNA methylation? DNA methylation is the process by which a methyl group is added to the cytosine site in a CpG site of DNA. This is mediated by DNA methyltransferase enzymes known as DNMTs. These use a methyl group from a S-adenosylmethionine, known as SAM, to methylate the cytosine. In general, there are two types of methylation. What we care about for this, when talking about somatic cells and cancer, is de novo methylation, which is specifically mediated by DNMT3A. So DNA methylation is a reversible process, while DNMTs perform the methylation of a cytosine at a CpG site. The 1011 translocation enzymes, specifically TET2, are responsible for demethylating. IDH1 and 2 are also involved in the processing of required resources for TET2. Additionally, Wilms tumor 1, which is a transcription factor, has been shown to recruit TET2 to in order to methylate sites in the gene that WT1 is targeting. This causes TET2 to demethylate nearby sites and then activate that gene. So what does methylation do exactly? So concentrations of CpG sites, referred to as CpG islands, are commonly found within the promoter regions. Promoter regions of genes that are actively transcribed are primarily unmethylated, allowing normal transcription. When these regions become more methylated, transcription is interrupted. or sometimes it can be stopped entirely. But this effect isn't absolute. Studies have found that the relation between DNA methylation and transcription is not one-to-one. Of interest, I mentioned DNMT3A earlier, which is the enzyme that methylates these sites. It's actually been found that when the promoter region of, one study found that when the promoter region of DNMT3A is methylated, it actually leads to the same effect that a mutation within DNMT3A would cause. So that kind of shows one of the possible effects in an interesting case. On a global scale, hypomethylation within coding regions leads to increased transcription, including genes that are normally repressed. Hypomethylation means that there's a reduced number of CpG sites that are methylated, but there still can be methylated CpG sites there. Causes of hypomethylation, DNMT3A mutations can lead to a hypomethylation phenotype. Sorry. Conversely, hypermethylation within the coding regions will lead to a reduction of transcription. Once again, hypermethylation refers to an overall increase of methylated sites, but it doesn't mean that none of the sites are unmethylated. So what does this matter in cancer? In cancer, it's been found that DNA methylation, and specifically differential methylation, can be used as a biomarker. Differential methylation is where samples in a group have markedly higher or lower methylation levels than expected, and this has been widely studied. Differential variability is still being investigated and it's not quite as established, but studies have shown the potential for both of these factors in specific genes to predict survival, relapse, and also disease progression. One study found that there was an association between high heterogeneity, meaning high variability of methylation status within a group, and their overall survival. In that case, it was their survival was lower. So how else is transcription modified? We've already talked about DNA methylation, so now we're going to talk about the other group, histone modification, chromatin modifiers. So as I said before, chromatin modifiers was number 3 in the AML genes in terms of how often it was mutated. So within this context, histone modification can affect transcription. More interestingly, histone modification can also affect the DNA methylation of sites near that histone. During histone modification, polycomb repressive complex 2, PRC2, is involved in the methylation of histones. When a histone becomes trimethylated, DNMT3A binds to the catalytic domain, EZH2, and subsequently methylates a nearby CpG. Additionally, ASXL1, seen here, affects these polycomb group proteins, especially PRC2. It's believed that ASXL1 can both activate and repress these PCGs. So that leads us to our study. What have from what we've talked about, we know that DNA methylation is a normal, essential process not specific to AML, and we know that AML patients commonly have mutations in these genes. So naturally, we led to this question: are DNA methylation and differential methylation and variability events more common in the genes of AML and AML patients with somatic mutations of DNA methylation genes? As I said, since DNA methylation is just an overall process, why are these so common in these patients? So for our study, our first aim is the development of an annotated integrated data set of AML patients from the Cancer Genome Atlas, TCGA. For this, we're going to combine the clinical methylation and mutation data sets into one data set, and it's going to require a lot of cleaning and harmonization, as well as extensive annotation to support studying our central hypothesis. Additionally, we're going to be evaluating the outcomes in TCA data so that we can analyze them in our second aim. Our second aim, identification of differential DNA methylation patterns in patients with mutated DNA methylation enzymes. So for this, we're going to be analyzing the differential methylation and variability within the AML genes, the cancer genes, and genes not associated with cancer. And our whole purpose behind this to see is to see whether or not these events are more common in any of those subsets of genes. Additionally, we're going to analyze genome-wide patterns of differential methylation variation. And at this point, once we've analyzed clinical outcomes, we're going to see if there's any association with these differential methylation and variation events with the outcome measures. So I'm going to describe the methods and the study design. The study is based off of the TCGA data, which is cases only. So for this, we're going to be, first we're going to be combining these two data. The data is found in two locations primarily. We're going to be getting the somatic mutation and clinical data from CBio portal, which is 194 subjects, and the methylation data from GDC data portal, which is 140. When these are combined, that comes down to 140 subjects. From there, we're also going to be doing an extensive amount of annotations so that these can be analyzed. So real quick, I want to talk about how we measure DNA methylation. This is a picture of the Illumina 450K methylation array, the chip for that. So within each of these 12 sample wells, there's over 450,000 sites that have a 450,000 probes. So when the DNA from the samples, from the different cells of the sample is fragmented, it is then bisulfite converted, meaning that any unmethylated sites will be converted to uracil, which is read as thymine, and then the methylated ones will be retained as a cytosine. So then they'll bind to the probe, and when a single base extension happens, it'll then send off a color signal. Red indicates that the site is unmethylated, and green indicates that the site is methylated. So as I said, each patient contributes one sample which contains many cells. This is then repeated across all 12 wells, and for each of those patients, it has an overall ratio of methylated to unmethylated. This presents a lot of issues when it comes to data cleaning, just because of the number of different factors that can influence it. A couple of those factors we need to account for is the different colors. There's a color bias inherent with the data in dealing with background noise. There's a number of methods that exist to deal with this, and so that's what we'll be using to remove the technical artifacts. Additionally, the 450K system uses two types of probes, which introduce another additional biases. Earlier I showed a demonstration of the probes. There's actually a little bit more to it, but we showed just the basic ones for simplicity's sake. We can see here that the different kind of probes kind of lead to a separation that needs to be dealt with. So how are these, as far as the annotation, how are these probes assigned to genes and genomic features? Primarily, we're going to be looking for probes that exist within transcription sites, so first exon, second exon, everything like that. The promoter sequences, and within enhancer sequences. We're going to be using the ENCODE database for assisting with enhancers and additional methods that help deal with just how complicated the enhancers can get with the chromatin. We have the support of the Beat AML project, which has dealt with these in the past, including Mike Mooney and Peter Rybin. So we have plenty of resources to help do this step. So since I mentioned before, this is a cases-only study. That means that we don't have healthy controls. So for this, what we are interested is looking at the difference in methylation between the samples that have mutations and the ones that don't. So what are the mutations that we care about? These are all the mutations that we're going to be primarily focusing on. These are the numbers you can see on the right indicate how prevalent they are within the TCGA data set. The first one, DNMT3A, as I mentioned before, is the methylation enzyme. The next group is TET2, IDH1, IDH2, and WT1. All of these are, TET2, IDH1, and IDH2 are all involved with demethylation, and WT1 has that association with demethylation as well. Additionally, we have two enzymes that are involved with histone modification. There are a number of other enzymes that exist within the PRC2, but we're choosing these because they've been shown in the past to be involved with AML and they're more common. Lastly, ones that I haven't mentioned before are FLT3 and NPM1. Neither of these are involved with methylation, but they are so prevalent in the AML data set that we will need to account for them. There's a couple interactions that we know that these have, specifically with DNMT3A, so we know that we can't just leave them out, but we do need to address them during analysis. So as far as the data goes, this is a co-occurrence plot showing how often these co-occur together and what our sample groups look like. Our ones that we will consider as our non-mutated samples are going to be these ones in green, which are ones that only have no mutations in any of these genes, have FLT3 mutations or NPM1. This comes to 70 samples. The rest in red are going to be the ones that are considered our mutated samples. This also comes to 70, so it's split 70-70 when just considering these. Now that being said, there's a little bit more complicated with this. I previously mentioned that hypermethylation within the promoter region of DNMT3A led to a similar effect downstream on the methylation. Because of this, we're interested in looking at that. So there's an extra little step in here where we're going to be looking for non-mutated samples that may have hypermethylation in their promoter or first exon that could lead to a similar effect downstream. So I've mentioned a lot about differential methylation variation, but I just want to touch on it a little bit more so it's a little more concrete. On the left, you can see an example of differential methylation. This speaks to the difference in mean between two groups. To identify this, we use linear modeling to determine whether or not there's a difference between two groups. As for differential variation or variability, this refers to a spread in the overall methylation status between samples. This is also going to be found using linear modeling techniques. So now that we have a better idea of differential methylation and variation, that brings us to the first part of our AIM2, where we're going to be looking at the genes within the different groups. As I mentioned before, the primary catalyst to why we came to this idea is that DNA methylation is common in all cells and not specific to AML, but it's very common in AML patients. So we're interested to see whether or not these mutations occur more common in any certain type of group. So all of the probes, which after cleaning there ends up to be just under half, so around 200,000 probes, will be grouped or rather annotated by what type of gene they also, or which of these categories they fit into. So the AML genes are going to be based off of a subset that Beat AML has already curated. Cancer genes coming from Cosmic, and then genes not associated with cancer. This is just a visual representation of the idea behind this. So in each of these groups, we'll have probes from both mutated and non-mutated indicated by the plus and minus. We're then going to compare the methylation between those, and from there we'll get a list of probes that have where we found differential methylation or differential variation. Then we're going to compare how prevalent these are within these groups and see if there's any difference. None of these probes will cross between these different groups. We can't do direct probe comparison, but we can see how often genes are hypermethylated, hypomethylated, anything like that within these different groups. After we do this, addressing our central hypothesis, we're going to then do a genome-wide analysis that's going to be more of an exploratory or, exploratory. So with that, we're going to then continue to do the methylation analysis across all probes and look for patterns there. The one issue is it's going to bring in a larger set of probes, and so it's going to incur a multiple testing penalty. As well, on the in our central one, we're going to be focusing on we're going to be looking at the clinical outcomes that are associated with differential methylation, differential variation. So as we said before, we need to evaluate all of the outcomes that exist in the TCGA data set and consider the effects of co-occurrence and also cytogenetics. and then we'll perform that analysis. So as far as limitations for the study goes, it's a relatively small sample size. It's 70 subjects in each group. Additionally, there's a large number of probes. As I said before, the array has over 450,000 probes per sample. And so there's a large multiple testing penalty with this. After cleaning, there's about 200,000, or just about half, probes left, and then from there they'll be further restricted in our first analysis based on ones that actually exist within genes. Lastly, there's a large co-occurrence of mutations, as showed in the figure before, with significant overlap. With that too, a lot of these mutations have different overall effects. Mutations in DNMT3A usually lead to a hypomethylation, while mutations in the demethylation genes lead to a hypermethylation. So there's concerns about how these effects could play with each other, and so that's one of our limitations. And additionally, we have concerns with FLT3 and NPM1, which we know are very common in AML, and also interact with other mutations that are present. So our plan for this is to do sensitivity analysis, but we are going to run into some issues with that due to sample size. As far as our timeline goes, this will be the primary research will be completed over the summer, and in the fall is when it will be completed. Thank you very much, and if you have any questions.
This is less about your methods, but more about kind of the output of this. Do you anticipate this would be something like diagnostic, prognostic? Is this something that you could just run in a simple panel, like a oncologist could just test for this? How do you anticipate your results would actually impact?
So I'll repeat the question. So the question was, what will the overall impact of this? Really, we're kind of trying to look more into. We've just noticed this pattern, and it's very interesting. And so we're trying to find out more about this, see if there's anything behind it. So it's kind of a pilot that could be expanded. Like I said, the sample size is pretty low, so I don't expect any groundbreaking discoveries, but that's immediately going to impact.
Yeah, just like in general, like looking forward to the future, but
yeah.
Not from an NF7.
Yeah.
Great talk. I'm curious to hear more about how you plan to incorporate prior knowledge in calling the somatic mutations for the chromatin and methylation modification genes.
So the mutations are already, the files that we're getting, it's all publicly available data from TCGA, and specifically with the mutation data they've already called and have given us the specific, they've given us what it is. Is that what you're referring to, or is it?
Yeah, I'm basically just asking, where you call mutations in each of these genes and how you've decided that they're true somatic mutations.
Okay.
Yeah.
So at this, I think we're just going to be taking TCGA at their word. So, because like I said, this is all publicly available data, mostly highly due to time constraints. So that's as far as we can really go on that. Okay.
So, just thinking about your outcomes. So what if you find that there is, for example, there's no difference between the, so what if you find that there is no difference in differential methylation between two of the groups, for example, the cosmic and the AML genes?
In that case, I would, that would lead me to believe that really it's not a, that would tell me that it may just be a fact that their cancer genes are maybe what's causing or leading to disease from there. That's one of the, I mean, if we find that out, it's still an outcome. It's still something that we'd be interested to know. We know, or, at least in this study, we wouldn't have found that there's overabundance of them within the AML genes, but it'd still be interesting for the field.
Okay, thanks.
Okay. Thank you very much. Yeah, right? Yeah, I kind of don't know that this because I think it's working, but okay, of course.
There's a lot improved from last time we saw it, so thank you.